back to the eye a little bit. So what you're looking at is a cartoon, and over on the left in the panel marked A, you see the pink cells. These are the retinal pigmented epithelial cells, and the important thing is that you can see that they literally hold and wrap around the photoreceptors, which are shown here in purple. In this case, we happen to be looking at rods. And so there's this intimate contact between the RPE and the photoreceptors, which is required to maintain the photoreceptor function and also its vitality. So the RPE has two functions. The first is shown down here in C. Don't worry about B, it's not relevant for us today. But the photoreceptors turn over all the time, and so our photoreceptors are being used, they accrue damage, and they're basically being rebuilt and recycled. And that's the province of the retinal pigmented epithelium. So if we look down here at the series of diagrams in C, what you're looking at is a cartoon version of the tip of the rod literally getting dumped off. And so, I'm sorry, the rods, the newest part of the rod are at the very bottom, which would be at the bottom of your diagram. And the oldest part of the rods, the ones that have been in service the longest, are at the top, and that's what I've marked with an arrow. So the retina has a very clever way of dealing with these old bits of rod, and it simply uses the RPE to encapsulate the tip and basically destroy it and get rid of it. And so this maintains the function and the youthfulness of the rods. The second really important function of the RPE is in phototransduction. And so we're going to have to talk a little bit about the retinoid cycle. So retinal, sorry, to get this diagram set up. So now you're looking at the RPE is shown here in pink and and it's been blurred or faded out to step into the background. The rods are shown here in purple still, and the rod, this first rod, is denoting a rod that is receiving light. So a photon of light hits the sensor, it's called rhodopsin, which is a protein plus the retinal, which is a light-sensing compound. A photon of light striking the retinal, and retinal is a vitamin A derivative, and so it's necessary to constantly replenish this from vitamin A. So when light strikes retinal, there's a photoconversion that changes the structure of the retinal. In order for a rod or a cone to begin to sense light again, it has to be reconverted back to the light-sensing form, and this is where the retinal pigmented epithelium comes in. So RPE 65, and just to step back 1 second, RPE 65 has one role, and that's to get vitamin A in so that we can make retinal, so that we have the photosensing compound to begin with. When light strikes this, it converts to all-trans retinal, which is fine, and then the all-trans retinal is converted into a retinol. All of this is occurring within the photoreceptor. Now, here's where the RPE comes in. So the all-trans retinal is now going to be bound by a protein called interphotoreceptor retinoid binding protein, or IRBP, and this is going to help transport the retinol into the pigmented epithelium where it can be regenerated. So RPE 65 is one of the functions of RPE 65 is to act as a chaperone to allow this process to go forward. And so once the retinol is in the RPE, then it's converted by a chemical process or an enzymatic process that we don't need to worry about. And then it's brought back, again, using IRBP, is brought back into the photoreceptors, completing the cycle. And so we have two proteins that we need to worry about, the IRBP and also RPE 65. And so this is a chaperone. And it's important, of course, for help transporting the retinal esters as well as getting vitamin A in, so we have the base. very different type of protein than PAC6. This is a protein that we can mess with very easily in the cell. And so the reason that RPE65 is so amenable to therapy is that, first of all, it's expressed only in the retinal pigmented epithelial cells, which, of course, are the cells that are located at the back of the RPE, which is located surrounding the retina. So it's expressed in one place and one place only. Secondly, it's a protein that has a very specific and a singular, or it has two functions, but the two functions are related to each other. And so you can put that protein in a synthetic form inside of a transport, which in this case happens to be an adenovirus. The third thing is, because it's only expressed in one cell type and it's expressed in cells that cells all the time, you can put a very simple control mechanism at the front end of this thing and have it drive. And so the bottom line is, this protein, RPE65, by virtue of the fact of what it does, where it's expressed, and how it does its job, is very amenable to using a gene delivery system such as an adenovirus, which is shown here schematically. And what happens is that we use the virus, and so this virus is infective, just like a cold virus or other types of viruses, except we make it in the laboratory. You package up your synthetic RPE65 coding region with your synthetic control, and you deliver it to the back of the eye, or you deliver it to the part of the eye that you want simply by microinjection. And once you get it there, then if you look to your left side, the virus does the rest of this. It infects the cells, it unloads its payload, and voila, you now have restored a functional copy of the RPE65 to the eye. This therapy works very well for this application. The problem is that PAC6 is too big, it's too complicated, and it has too many layers of control to allow it to be packaged into a virus easily. And so one of the concerns that I have about using a viral delivery system for PAC6 is that, yes, you may fix the problem for a short while, but the danger is that you may, because you're overriding all of the normal cellular control mechanisms for PAC6, that you may get very unintended consequences. So what are some of the unintended consequences? If we go back to PAC6, where PAC6 is expressed. And so if we go back to this image for just a moment, it's not the perfect image to set this point up. But PAC6 actually, it'd be better if I could get okay, we'll stay with this one for a moment. What you're looking at, again, is the message where PAC6 is expressed. And so PAC6 is doing many different types of things in the body. One of the things that it's doing in the brain, and probably also a little bit in the pancreas, and certainly in parts of the eye, is controlling cell proliferation. Too much PAC6 is as bad as having not enough PAC6. And so the danger with a delivery system that's no longer regulatable, such as an adenous system, is that the cells are going to have too much PAC6, and this can lead to unintended consequences such as tumor formation or other problems with changing the cellular machinery. We don't know that that's true, and it's certainly something that we're messing with. But PAC6 as a gene does not lead itself as well to this approach, an adenovirus approach, as does something like Lieber's RPE65 and some other proteins that have very specific functions in cells. And so I think with that, we've been going a little bit longer than I expected, so I will stop and take any questions that you might have.
So you talked about that this approach wouldn't work, or you don't know that it would be the best approach for PAC6. What is an approach that you would be looking at to deliver, I guess, a gene therapy?
Sure, that's the obvious and good question. So, I think so. I don't want to completely rule out adenovirus approaches for PAX6 because we certainly can use them in a limited setting, um, but it will not cure aniridia in the same way that you can cure Leber's. Things that we can do are to develop new cell-based therapies, so basically stem cell-based therapies in which we fix the PAX6 gene in the cell type, and that's one of the things that we are pursuing with the cornea. That's very amenable to taking up large pieces of DNA where we know that the regulatory mechanisms are preserved. It's a very difficult approach. One could, in principle, use an adenovirus delivery system to deliver PAX6 to certain types of cells, culture those cells, make sure they're doing their job, and then put them back in the person. But these are all things that would involve some sort of surgical intervention at some level. The real answer is it would be better to have prenatal screening. Conceptually, it is possible to go in and begin to genetically engineer somebody so that you repair the PAX6 mutation right away. If you know what the mutation is, you can go in and fix it in a recently fertilized egg. Clearly, that has its own attendant problems because now you're into eugenics. The one approach that one can take right now is in vitro fertilization. So again, if you know what the PAX6 mutation is, you can screen for fetuses that either carry the mutation or that don't carry the mutation. This approach is using existing technology, and of course, it's been in wide use now for a variety of genetic disorders for almost 20 years, and so the technology is very well established. Not a particularly satisfying answer, but it's at least those are the things that are on the drawing board right at the moment. Any follow-up questions to
But
that?
the answer there is more you're selecting prior to having a child with aniridia. What are some approaches to dealing once somebody already has aniridia? So in the Leber's, they took the virus, and when you can see it going in with a needle, how would you deliver some sort of therapy to the different, I guess, components of the eye with somebody in aniridia, just theoretically?
Okay, so theoretically, one could take a similar approach to what is being diagrammed here for Leber's. The other significant difference is that because PAX6 affects the development of all parts of the eye, essentially what we would have to do is to come up with a new way to completely grow part of the eye. And that's not been done yet, although a lot of people are trying. So at best, what the approaches that are best, the approaches that are available to people are going to be some combination of gene therapy, cellular-based therapies, and some sort of surgical intervention. In Leber's, what you're doing is you're fixing a cell that's not doing its job. In PAX6, the cells simply do not exist, and there's no way at the moment to begin to rebuild or regrow the retina in a way that is necessary to repair that damage.